Today we will discuss the next intestinal parasite, that is Taenia. Before entering into the details of Taenia, let us first recall the classification of parasites so that you are clear regarding which class of parasites we are discussing. Parasites are divided into protozoa and metazoa. Protozoa are then divided into four, that is amoebas, sporozoans or epicomplexa, flagellates and ciliates. If you remember, we have already covered the protozoal intestinal parasite, that is amoebiasis and giardiasis. Now we are moving to helminths. Helminths are divided into platyhelminths and nemathelminths, that is the nematodes. Platyhelminths are then again divided into trematodes and cystodes. Today we would be discussing Taenia, that comes under the class of cystodes, that is the tapeworm. Let us have some brief introduction regarding the class cystodes. Cystodes comes from the Greek word kystos, which means ribbon, and as the name suggests, these worms are ribbon shaped. Cystodes are long, they are segmented and they are dorsoventrally flat, that is they are tape like, and hence they are named as tapeworms. Based on the habitat, cystodes are classified as intestinal cystodes and somatic or tissue cystodes. In intestinal cystodes, the adult worms they inhabit in the human intestine, while in somatic, the larval form of the cystodes are found in human muscles or organs. Examples of intestinal cystodes are Diphyllobothrium latum, Taenia solium, Taenia saginata and Hymenolepsis, while somatic cystodes are Taenia solium and Echinococcus granulosus. We will see in detail Taenia solium and Taenia saginata. In general, the helminths they exist in three morphological forms, that is the adult form, larval form and eggs. Now let us see the adult form of Taenia. As you can see in the picture, this is the adult worm of Taenia. It is long, it is segmented and dorsoventrally flattened, that is it is tape like. Now the adult worm is again consisting of three parts. The first is the head, which is also called as the scolex. As you can see here, the scolex of the adult worm. This head is the organ of attachment and it helps in attachment with the intestinal mucosa, and this attachment occurs with the help of these four cups like structures that are also called as suckers. The next part is the neck, from which the proglottids arise or from which the body starts. And the third part is the trunk, also called as strobila or the body of the Taenia. The body of Taenia, it consists of number of segments. As you can see here, number of segments. These segments are also called as proglottids, and the length of any tapeworm depends upon the number of these segments. Now these proglottids, as you can see in the picture, can be immature, can be mature proglottids, or they could be gravid proglottids, wherein the eggs of Taenia lies. Also remember that cystodes are hermaphrodites, also called as monoecious, that is they contain male and female organs in the same segment. Then the next is the eggs. The eggs they fill up the gravid segments, and in most of the cystodes the eggs are round to oval It consists of 3 pair of hooklets. It also has a radially striated embryophore and a very thin eggshell, also called as hexacanth embryo. As you can see in the picture again, the head part which has got the suckers, then comes the body, the immature segments, the mature segments and the gravid segments. As you can see the detail of the gravid segments. In the gravid segments, the uterus after fertilization get filled with eggs. A clear picture of the eggs of Taenia. As you can see, they are round to oval in shape. They have got 3 pair of hooks, a radially striated embryophore and a cyst wall or the outer coat. Also, these eggs are bile stained. We have already seen the basic structure of the forms of Taenia. Now let us see Taenia saginata and Taenia solium individually. Starting with Taenia saginata, the head of Taenia saginata has got 4 suckers which helps in adhesion to the intestinal mucosa. The head, that is calyx, is around 1 to 2 mm in size and it has got no hooklets. The adult form of Taenia saginata measures around 5 to 10 meter in size and it has got around 1000 to 2000 segments. So you can see here it has got around 1000 to 2000 proglottids. And this is the picture of the gravid proglottid of Taenia saginata. As shown in the picture, the central longitudinal stem of the uterus from where this dichotomous branching is seen. Then the egg of Taenia saginata measures around 30 to 40 micrometer. And the third form, that is the larval form. The larval form of Taenia saginata is called as the Cysticercus bovis. Now let us see how the life cycle of Taenia saginata works. The life cycle of Taenia saginata is complex, that is, it requires 2 hosts, definitive host and intermediate host. Definitive host is the one in which the parasite undergoes sexual multiplication and intermediate host is the one in which parasite undergoes asexual multiplication. The definitive host in this case is human and the intermediate host is the cattle or cow. And the mode of transmission is ingestion of contaminated or undercooked beef. Now when humans, they ingest this undercooked beef which is contaminated with the larval form of Taenia. When humans eat this contaminated beef, it enters into the GI tract of intestine and the larval form which was there in the muscular portion of the beef, it evaginates and in the small intestine and attaches itself to the mucosa of the small intestine. This then develops into the adult worm. As you can see here, the larva develops into the adult worm and it undergoes self-fertilization within the segments to produce eggs, and these eggs are formed inside the gravid proglottids. Now these gravid segments or eggs of the Taenia, they pass in the feces and the eggs are then consumed by the cow while grazing on grass. This cow, when ingest the eggs of Taenia, The eggs penetrate the intestinal wall, enters the bloodstream and enters into the muscular portions of the animal, wherein it develops into the larval form, that is the Cysticercus bovis. And again, when this beef which is undercooked or contaminated is ingested by humans, the cycle continues. Moving on to Taenia solium, it has got some differences from Taenia saginata. As you can see in the scolex, Taenia solium, it has got a rostellum with 2 rows of hooklets, and hence it is also called as the armed tapeworm, as it has got hooklets. It also has 4 suckers. Then the length of Taenia solium is around 2 to 8 meters, and the number of segments are around 800 to 1000. And the gravid segment, as you can see here, there are fewer numbers of uterine branches than Taenia saginata. And this is the larval form of Taenia solium, which is called as Cysticercus cellulose. Life cycle of Taenia solium. It has got a complex life cycle with definitive host as humans and intermediate host as pigs, in contrast to the intermediate host of Taenia saginata, that is the cow or cattle. In this case, the mode of transmission is ingestion of contaminated or undercooked pork. So when humans ingest undercooked pork which is contaminated with the larval form of the Taenia solium, that is Cysticercus cellulose, the Cysticercus cellulose contaminated pork enters into the GIT, and in the GIT the scolex, they evaginates and attaches to the intestinal wall, matures to develop the adult worm, undergoes self fertilization and lays eggs inside the gravid segment. This gravid segment might then rupture in the intestine or perianal region, or when it comes outside to the environment. Along with the gravid segments, the eggs might come into the environment via the feces. And when these eggs are then consumed by pigs, the eggs then enters the GIT, penetrates the intestinal wall, enters the bloodstream and finally enters the muscles of pig. And when this contaminated pork is ingested by human beings, the cycle continues. As you can see in the picture, the contaminated pork, contaminated with Cysticercus cellulose. Now in case of Taenia solium, unlike Cysticercus bovis, which is the larval form of Taenia saginata, Cysticercus cellulose can develop in humans. And in this case, the infective form of Taenia solium to humans is the egg. These eggs of Taenia solium might reach the GIT of humans via 3 routes. The first is consumption of contaminated water or vegetables. The second is auto-infection by unhygienic habits. That is, I already told you the gravid segments, they might rupture at the perianal region, and because of some unhygienic habits, through the feco-oral route, the eggs can be ingested. And the third route is the reverse peristalsis in human intestine. These gravid segments, because of reverse peristalsis, can enter into the GIT. Now once these eggs are entered into the GIT, The liberated embryos then penetrate the mucosa to reach any part of the body through blood, and these eggs then develop into the larval form, that is the Cysticercus cellulose. And the sites at which Cysticercus cellulose are formed are subcutaneous tissue, muscle, brain or eye. This Cysticercus cellulose then cannot develop further, and hence human infection is a dead end for this larva. When this Cysticercus cellulose enters the brain, it causes neurocysticercosis, and this we will deal later in CNS infection. Epidemiology of Taenia. As the intermediate host of Taenia saginata is cattle or cow, it is more common in the cattle breeding areas, and the areas with the highest prevalence include Central Asia, Central and East Africa, while Taenia solium is endemic in Central and South America, Africa, Southeast Asia and Philippines. Clinical manifestations of Taenia. When it comes to intestinal taeniasis, majority of the human beings are asymptomatic. They only come to know about the infection when they feel a discomfort while the passage of proglottids in the feces. The general symptoms might include mild abdominal pain, nausea, loss of appetite. When the proglottids pass through the feces, patient might get some perianal discomfort and pruritus. And in case of Taenia solium, when the Cysticercus cellulose infects humans, it might cause cysticercosis, which depends upon where is the localization of Cysticercus cellulose. You can see the picture of cysticercosis in eye. A picture of neurocysticercosis, that is the Cysticercus cellulose localized in the brain. Laboratory diagnosis of Taenia. Stool examination is carried out for the demonstration of proglottids and the egg forms in the stool. To increase the detection rate of stool examination, multiple stool are collected and also concentration techniques can be applied. The egg forms of Taenia saginata and Taenia solium are mostly similar, except that the eggs of Taenia saginata, they are acid fast, while the general look of egg of the Taenia, as we have already dealt earlier, the egg is around 30 to 40 micrometer and it consists of an embryo with 6 hooklets. Also, these eggs are bile stained and they do not float in saturated salt solution. And the other form that we look for in the feces are the proglottids, and for the differentiation of proglottids of Taenia saginata and Taenia solium, lateral branches in the uterus are to be seen. The other methods available for laboratory diagnosis are antibody detection, Taenia specific antigen detection in stool, biopsy in case of human cysticercosis, imaging techniques and molecular techniques. Let us see few differences between Taenia solium and saginata. As we earlier saw, the length of solium is 2 to 8 meters while saginata is 5 to 10 meters. And the scolex or the head of the solium has got 2 rows of hooklets while saginata does not have hooklets. There are 800 to 1000 segments in Taenia solium and 1000 to 2000 segment in saginata. And the branches in the gravid proglottids are 7 to 12 in case of Solium and 15 to 30 in case of Saginata. You can see the difference, rows of hooklets, while here there are no hooklets in case of Saginata. The lateral branches in the uterus, you can see lesser number of lateral branches in Solium and more number of lateral branches in Saginata. For prevention of intestinal taeniasis, adequate cooking of the beef or the pork is required. Thorough cooking at 65 degrees Celsius for 5 minutes is adequate, and refrigeration at 4 degrees Celsius. There are other methods for the prevention like meat inspection and effective fecal disposal so that infection to cattle and pigs can be prevented.